Okay, so yesterday we went over all the parts of the cell and what they do. All right, so you can match that up with your cell diagram. And then we talked about 2 out of the box and 3 out of the big 3. We kind of just got to active transport at the end. Okay, so diffusion, osmosis, and active transport are what I call the big 3 methods of cellular transport. Okay, so just as a quick review, diffusion is the passive movement of any material down the concentration gradient, or from high concentration to low concentration, the way they would naturally move. Okay? Osmosis is the movement of water only across a membrane in order to gain an equilibrium for salts, okay, or any dissolved solutes. Okay, so it's only water that moves across in osmosis. Okay, there's salt on either side, but it's not equal in concentration, so water moves to the side with the higher concentration in order to dilute it and try and make it equal. Okay, we talked about how that's why a slug will shrivel up, or a leech will shrivel up, okay, if you put salt on it. Anyone ever had a leech on them? Okay, if you have, that's how you get them. Well, I mean, you can tear them off, but it's better to just put a little salt on them. They generally let go, okay, right as soon as you put the salt on, because they have the same reaction to it that slugs do. Okay, water starts to come out of them, and they actually, it's not even really voluntary for them. They just automatically let go when that happens. Okay, so that's osmosis. And then active transport is the only one of the big 3 that requires a cell to use energy. Okay, so in active transport, the cell uses energy in order to cause the carrier proteins embedded in its membrane, and I underlined that yesterday, said that's an important one. Using energy was important. Okay, it causes the carrier proteins to change shape and open and close, causing a pumping action to occur that will pull, okay, materials from an area of low concentration to an area of high concentration, which is the opposite way they generally want to move by nature. Okay, so active transport expends energy to basically pump water uphill. Okay, it's moving things in the direction they wouldn't normally go. All right, is that ringing a bell for everybody? Okay, so the key things here, guys, the way I'm going to ask this on your unit exam is like this. I will give you a real world situation. Okay, and I will describe what's going on, and then I will say which cellular transport method would be most involved in this situation. Explain how that method works and why it applies here. So if I, was to say you had a leech on your leg and you put salt on the leech and the leech shriveled up and let go, which cellular transport process would be most involved and how does it work? Okay, then you would say to me, well, this seems like a situation where osmosis would most apply, because osmosis tries to balance salt concentrations by moving water across the membrane towards the area where salt is more concentrated, attempting to gain an equilibrium, and this would cause the leech to let go of you because it would shrivel up. Okay, something like that. Does that sort of make sense, what I'd be looking for? Okay, you'd first off identify that this was in fact osmosis. You tell me how osmosis works, how it applies to that situation, and what would happen. Okay? Okay, so that's what I'd be looking for. It'd be a 5-mark question, so you'd want to make sure you explained it pretty well. You wouldn't just say osmosis, because that gets slugs to come off. Okay, that might be 2 marks at best out of 5. Okay. All right. Now we got to talk about methods of transport in and out of the cell, but that involve changing the membrane as opposed to moving stuff through it. Okay, because osmosis and active transport move stuff through the membrane. The membrane doesn't have to change shape. Okay, in exocytosis and endocytosis, the membrane actually has to physically change because whatever's being transported is too big to go through it. Okay, so this is how a cell will take in large materials or just large amounts of materials, because the 3 we've talked about don't move huge amounts of materials, and they definitely don't move large materials. Okay, like for example, the cell does not use active transport to eat. Okay, if we're talking about an animal cell, it does not actively transport food particles through those little carrier proteins. They're way too small. All that goes through those carrier proteins is things like sodium, potassium, so like single atoms, maybe small molecules, that's it. Water, okay, that can go across the membrane simply because the membrane is, structured so that water can go across it, okay, or actually through it, rather than for it to have to be engulfed. Okay? Not that the membrane doesn't engulf water from time to time, but most of the time it just goes across. Okay? So these ones that we're looking at involve big stuff going from the inside of the cell to the outside, or from the outside to the inside by changing the shape of the membrane. All right, so transport out of the cell we call exocytosis. Okay, and that's, remember I said when you're giving me the function of the cell membrane, I wanted two words, endocytosis and exocytosis. That's what the membrane does. Okay, so exocytosis, the cell secretes, or we could also say excretes if it was waste. Okay, large molecules by having the vesicles, so those little bubbles we talked about with the Golgi, okay, merge with the membrane and then pop outwards. Okay, that's how exocytosis works. So that the Golgi apparatus packages some material, wastes or hormones or whatever, that package goes to the membrane, and when it touches the membrane, because they're made of exactly the same material, it just becomes part of the membrane, which ends up popping this, and whatever was inside of it, these blue bars, is forced out into what we call the intercellular space, okay, or interstitial fluid, which then usually is in contact with the blood. Okay, so that's the process of exocytosis. It is the same regardless for what is inside the vesicle, whether it's wastes or hormones or whatever. This is what exocytosis looks like. The bubble comes off the Golgi, it goes to the membrane, it merges with the membrane and pops the stuff out. Okay, so that's different from the big three because the membrane actually has to change, whereas in the other three the stuff just moves. Okay, everybody all right with exocytosis? All right, so there's only one way that stuff gets out, and it's by this. There are actually three different forms of endocytosis. Okay, so three different ways that things get into the cell by changing the shape of the membrane. Okay, so three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay, those are the three ways that the cell can basically engulf stuff. And mechanically, all three are the same. The membrane just envelops something and it gets inside. The only reason we consider them to be different is because of what they take in when that happens. Right? So that's what we'll look at here now. Okay? Now, for a cell to eat, like especially the amoeba, this is how the amoeba eats. Okay? It uses a process called phagocytosis, which is the enveloping of a material, okay, and just taking it into the cell in a vesicle or a vacuole. Okay? Usually if it's got food in it, we call it a food vacuole. Right? So the membrane just kind of does this. Anybody have to walk their dog and clean up after their dog? Same process. Okay, so if a cell wants to eat something, okay, so let's say that, right? The cell just does this. It just envelops it, turns the membrane inside out. Now it's enclosed in its own vesicle inside the cell. Okay, pretty simple process. Exocytosis is the opposite. This comes along, merges with the membrane, and pops it back out again. Okay, everybody all right with that? So that's all it does. The membrane just envelops the material, okay, and allows it to come into the cell. Okay, so phagocytosis, the cell engulfs a particle by wrapping what's called a pseudopod, which is called a false foot, okay, just envelops it, okay, around it and packaging it within a membrane-enclosed sac large enough to be called a vacuole. Okay, this is also how a lot of your immune system's cells kill infection. Okay? You have a lot of immune cells that will actually eat invading organisms in order to get rid of them. Okay? All right, so phagocytosis is engulfing a solid particle. Okay, so the membrane engulfs a solid particle, usually food. Okay. Pinocytosis works mechanically the same, it's just instead of enveloping food, it envelops water. Right? Cells don't usually do this unless they're severely dehydrated. Okay. So this is an electron microscope picture of a cell undergoing pinocytosis. Okay? You can actually see all the spots where the membrane is engulfing fluid. Okay? So if a cell becomes really stressed in terms of hydration, and then suddenly there's fluid available, it'll just start doing this. It's like chugging. Okay? It's just chugging water like crazy. That's the equivalent of that for a cell. Okay? It's exactly what you would do if you were really thirsty. You just pound water. Okay? Well, this is how a cell pounds water. Okay? That sort of makes sense? Right, but again, the mechanism is exactly the same. The membrane just folds in around it and envelops it. Okay. And the last one is receptor-mediated endocytosis. So if you recall the other day, I said your cell membrane's covered in all kinds of stuff. And sometimes, some of those things are receptors. So there's like little antennas that are on the surface of the cell membrane. And they receive certain chemical signals. So if one of them is for adrenaline, and an adrenaline molecule lands on that receptor, the cell will automatically engulf it and bring it into the cell so that it can cause the changes in the cell that it needs to change. Okay? That gets it in quickly, because typically hormones are too big to move in by active transport. Right? So they are just engulfed instead. Right? So on the surface of the membrane are these receptors. The receptor gets triggered and it engulfs that stuff. This is actually what it looks like under an electron microscope. Okay, so you can actually see some of the receptor molecules here. And then in here, okay, you can see this is like kind of the cup shape, okay, that they would have. That's not what I wanted. Okay, this kind of cup shape here where they've received the signal, the cup is full, okay, and so that cell membrane, this is the cell membrane right here, has engulfed it and it's about to close off, okay, and have that stuff come in. Right? So receptor-mediated endocytosis is caused by a chemical signal telling the cell to engulf the material. All right, questions on those? Okay, so these three that involve changes in the cell membrane are not as important as the big three. Okay, so in other words, I'm never going to ask you a written response question about these. Okay, you might want to write that in your notes. These three, phagocytosis, pinocytosis, and receptor-mediated, will never be a written response item. They will be a multiple choice item, okay? But osmosis, diffusion, and active transport will all three likely be written response items, okay? Because they are more important and happen more often. Okay, questions on those? I would like you guys to answer those 4 questions. I'll give you a few minutes here to work on them. Okay? Give complete sentence answers if it's appropriate. Okay? And then we'll talk about them, and then we'll start looking at the cell anatomy lab that we're going to do on Tuesday. Okay, let's have a look at these. So, color blindness is caused by your cells being unable to produce certain proteins. Where's the problem? Because it's certain proteins, it's probably not the ribosomes. If the problem was with the ribosomes, your cell wouldn't be able to produce any proteins. Okay, so it's the certain part that kind of eliminates ribosomes as the problem. Okay? Where do the ribosomes get the information to make proteins from? Yeah, from the nucleolus, which gets it from your DNA. Color blindness is genetic, okay? And it comes from a mutation of your DNA. Okay? That mutation makes it so that the protein your cells produce is not quite right. Okay? It's, what is it, rhodopsin or something like that. It doesn't produce it quite right. And so some of the color receptors, the cones in your eyes, are unable to detect certain wavelengths of light. Right? Most people don't even know that they're colorblind until they take their driver's test. Okay? Where you have to look at these like patches of dots, and they're slightly different colors. And so they form a pattern like a number. And if you're colorblind, you can't see the number, okay, on some of these little palettes of dots. Okay? Mr. Van Erb, who subs here sometimes, okay, he used to work here. When he retired, he's completely colorblind, like the worst case of colorblindness I've ever seen. Right? He is completely colorblind. When he retired, we bought him the glasses that allow colorblind people to see colors. It was, when he put them on, it was like somebody smacked him in the face. Okay, like he put these things on, it's like, that's green? Like he had never, ever seen green. He's like severely red-green colorblind. Can't see green, okay? Because his genes don't have the protein that allows the cones to see green, okay? So he had never, like when he looked at trees, he just saw something else. Now, again, it's not really that bad for him. He just doesn't, I mean, he kind of knows, he understands that trees are green, but he doesn't see the green that you or I would see. Okay, but it's quite a big deal, okay? Because we would sometimes play tricks on him. Okay, like one day he and I were wearing an outfit that was very, very similar. I was wearing blue pants and a shirt that had a checkered pattern on it that was blue and like burgundy or something. Okay, he was wearing black pants with a checkered shirt. It was like black and yellow or something, but it was exactly the same pattern. And he's like, hey, look, we're twins. And I looked at him, yeah! And I took him down the hallway, look, everybody, we're twins. And everyone's like, what's wrong with you guys? He didn't know. He thought we had actually accidentally worn exactly the same outfit. I'm not saying that was like, we're good friends, so like he knows that we give him a hard time about his color blindness, and so it was funny in that way. I wouldn't do that to someone I didn't know, because that would be mean. Okay? But he can't see colors. All right? Now, is that a problem if you're a driver? Well, we sort of think it is, but now think about how stoplights are set up. Green lights are always where? Bottom or right. They're always on the bottom or the right of the tree, right? So if they're arranged horizontally, green's always on the right, okay? And green is always on the bottom if they're arranged vertically. And with the flashing arrows, okay, it's flashing if it's green, but once it goes yellow, it's solid. All of that is around color blindness, okay? Because not everybody can see those colors the same way. For Mr. Van Erb, it's not a big deal the difference between the red and the green and the yellow lights. He knows what the position is on the tree. But the flashing yellow, the flashing green and the solid yellow, if they didn't have a pattern difference, okay, like the flashing versus solid, he wouldn't know. And he would go through a solid yellow arrow, thinking it was green. And that's why the arrow flashes when it's green, but is solid when it's yellow. And that way people know, okay, it's yellow, even if they can't detect the difference between green and yellow. Because Mr. Van Erb can't. He looks at the arrow, he sees the arrow is lit. That's it. Okay? So there's a reason why those things work the way they do. And if you ever wondered, you probably didn't even really notice up until now, but that's why they actually work that way. Okay? In the same way that, have you ever noticed that coins all feel different around the edges? Okay? Like a quarter's got ridges, a loonie's got edges, and on a toonie, it's rough, smooth, rough, smooth, rough, smooth all around the edges. Okay? That's so people who are blind can tell what coin they have in their hand. They just run their fingers along the edge. Okay? If it's small, like really small and rough, it's a dime. If it's a little bit bigger and smooth, it's a nickel. Okay, if it's a little bit bigger and rough all the way around, it's a quarter. Okay, if it's a little bit bigger and like edged, it's a loonie. And if it's a little bit bigger, rough, smooth, it's a toonie.
A Canadian white has brown on it too, right?
The paper money.
Yeah.
Yes, new. That's new though. It didn't always. Yeah. Have braille on it. But yeah, that'd be, yeah, if you were dealing with paper money, that would be hard as a blind person for sure. Or you just take out your card and tap. But, okay, then it's hard because you can't tell what the thing says, right? People might be dishonest with you and tell you it says something different on it. All right. Question number 2. Why is it important for organelles to have their own membranes? This comes back to that big long word that starts with C. Compartmentalization. Each organelle needs its own specific environmental conditions in order to do its job properly. And that is why we have compartmentalization and why each organelle has its own membrane. It will enclose its own separate environment. Okay, we talked about number 3 yesterday. If a lysosome's membrane ruptured, what gets out?
Digestive enzymes.
Yeah, digestive enzymes will get out and they'll digest the cell from the inside, so that'd be bad. Okay, and number 4, what problems might a cell have if all the ER was removed? This is incidentally how I sometimes backhandedly ask, what is the job of this organelle? What would happen if you didn't have any of this? Okay, is the kind of backhanded way of saying that. Right? So if you didn't have any rough ER, you wouldn't be able to transport proteins. That's kind of a problem. If you didn't have smooth ER, there'd be a lot of things your cell couldn't do. It wouldn't be able to repair membranes, wouldn't be able to detoxify poisons, break down complex carbohydrates, or make fat-soluble hormones. Okay? So it would probably die. Okay? Now, if that's a written response question and you write, it'll die, and it's a 4-mark question, I'll give you 1 mark for that. If you say, it'll die because it can't do these things, then I give you 5 marks for that, or 4 marks, whatever it's at. Okay? So that's the difference. The level of detail is really, really important in biology. Okay? Tell me everything you know when you're telling me about something. All right, questions on the questions, or the answers. Okay. Then, let's have a look at the cell anatomy lab here. Okay, I don't have the Chromebooks here, so you can pull it up on your phone if you want to, or you can just follow along with what we're doing here. Okay, so our lab on Tuesday, we are now going to be looking at cells for the purpose of identifying what structures are necessary for them to carry out their basic functions. You know, that whole second point of the cell theory thing. Okay? What structures are necessary for a cell to carry out its basic functions that we could at least observe with a light microscope? Okay? So to that end, we are going to look at a typical plant cell, a typical animal cell, right? And I'll also have a human blood smear there, but it's not like super important for you to look at it. It's just kind of there because everyone always asks about it after they look at the animal cell. Okay? So those are going to be our important slides. Now, when we start looking at these things, okay, our plant cell slide, and it's going to have that circular cover slit cemented on in the middle like the ones you used the other day. And on it, there will be 2 little V-shaped things like this. They're really thin though. Okay, like really you have to hold it up to the light probably to see them. You need to look at the bottom one. And you want to look at this part. Okay, either here or here. Don't look at the middle and don't look at the top. Okay, because it's got a leaf from a monocot, which is a type of plant, and a leaf from a dicot, which is also a type of plant. And we want to look at the dicot leaf, which is on the bottom. Okay, now, these look weird, right? Like you're looking at this and going, a leaf does not look like that, Coder. Well, it does if, sorry, plant. It does if you look at it this way. Okay, so really what's happened is the plant's leaf has been cut like this, and we're looking at it in cross section. Okay, so it's just a very thin slice of the leaf from this direction. Okay, so we want to look at the parts of the leaf that are kind of right here and here. So when we're looking at it on the microscope slide, that will be on the downward sides of those little triangles. Okay. Now, when we look at that, this is what we're going to see. Okay, this here. So you're going to see a bunch of different layers. Okay, you're going to see this layer here on the top that looks like they're just kind of big and transparent, although you can see a nucleus in some of those. Okay, and then there's going to be these layers that are kind of like, these are the typical ones. These are the ones we want to do our diagram of that are kind of rectangular. And then there's some down here that are kind of randomly shaped. Okay, and then there's another layer of the clear ones on the bottom. Okay, we want to look at this layer here, which is called the palisade layer. Right? Can you see already some pretty obvious structures? Okay. What would this thing right here be? What part of the cell would that be? All right, okay, let me do this. So here is the plant cell. Okay, that's one plant cell. What's the pink thing?
Nucleus.
That's the nucleus. Okay, so that's the nucleus. Inside of it is a darker dot, which would be the nucleolus. Right? And if you look closely here, kind of on the top, see how it's kind of irregularly shaped? It almost looks like a bit of a triangle at the top there. What's always attached to the nucleus? I told you it sometimes looked like the nucleus has a beard.
ER.
Yeah, that's the rough ER. Okay, just giving you an idea of what these things look like for real, right? Because they don't always look exactly like our diagrams do. Okay, what do you suppose all these green things are? Oh, the chloroplasts. Remember I told you they're actually quite large and fairly obvious. Now this is enlarged a little more than it's going to be in your microscope, but okay, those are the structures that you're going to look for and are quite easily visible. Okay. Through the microscope, it's going to be probably more like this. Come on. Probably more like that. Okay, so quite a bit smaller. Okay, your animal cell is going to look like, oh sorry, we're doing 4 slides, sorry. Okay. Your animal cell is going to look either like this one or like this one, depending on what dye pattern you get. Okay. Come on already. Okay, you see all the little purple circles? Each one of those is a nucleus. Okay. So if I was looking at, let's say, this nucleus right here, this would be the cell that it's in. Okay. The animal cells are smaller than the plant cells. Okay. So that's kind of what one of the dye lots look like, but the other dye lot looks like this. Okay. So there's a nucleus, and there's the cell it's in. Kind of looks like an eye. Okay. When you look at that, you can see that the edges of the nucleus, especially right there, are a bit fuzzy. That would be ER. Okay. You'd probably be looking at like maybe a Golgi right there. Okay. Certainly you can see the cell membrane. You can see the nucleolus. I'm not saying use your imagination when you do your labels, but to a reasonable level, use your imagination, because you kind of have to a little bit. Okay? I'm not saying like guess, but it's reasonable to assume that those would be the structures we'd be looking at. Okay? All right. We're also each going to make a slide of our own cells. Okay? And we're going to take them from your cheek. Right, so you're going to get a Q-tip, and you're going to swab the inside of your cheek with it, okay, like they do on CSI, and you're gonna get slime on there, okay, and then you're going to smear it onto a microscope slide, like we did, like we put the letter E on, you're just gonna kinda go, so if this is my slide, okay, and now my pen is the Q-tip, okay, I'm just gonna go back and forth, and I'm gonna twist, so I get all the sides of the Q-tip smearing on there, okay, make a nice good smear like that in the middle, put a drop of water on there and a cover slip, and you've got a slide of your own cheek cells, okay. Your cheek cells will look like this. That's cheek cell, that's me, that's my cheek cell, okay. You can see nucleus there in the middle, okay, so right here would be the nucleus, right, and I would say that probably that bottom little bit there would probably be the rough ER, okay. I would say that we'd probably be looking at maybe lysosomes here and there, okay, and this is probably a Golgi right here, okay, and might have a peroxisome or some other ER over here, okay, something like that. And again, you can be reasonable with what you think things are. All right, this is another version of my cheek cells. This is where I just actually got quite a few in that collection there. Okay, and you can see nucleus and the cell membrane and all that. If you see something that's quite dark and large, it's food. Okay, no matter how well you brush your teeth or mouthwash or whatever, you're always going to have bits of food stuck to the inside of your mouth. And they're going to come off on the Q-tip, and they're going to be visible under the microscope, and no one will make fun of anyone else, okay, for their oral hygiene, because everybody will have some of those in there. Just know that if you see something big and dark, it's not one of your cells, because your cells are going to be almost transparent, okay, because they're not dyed, okay, and they're very thin. Right. And then we'll also have human blood to look at. I'm not even worried if you, I don't even really want a diagram of the human blood. It's just something for you to look at. Okay. These are all red blood cells except for these. These are eosinophils, which are white blood cells that are part of your immune system. All right, everyone kind of understand what we're going to be looking for? All right, so like we said, the problem that we're going to be investigating is that cells have to carry out the basic functions of the organism in order to survive, so they have to have structures within their cells to carry them out. So your problem has to be in the form of a question. What structures does a cell need to carry out its basic functions, or something along those lines would probably be good. Okay, hey, I'm doing that part where I give you the big juicy hints, by the way. Okay. In this lab, we're comparing cells from the two modes of nutrition. We are looking at photoautotrophs and chemoheterotrophs. Okay, we're looking at one plant and one animal. Right? That's the way we have to do it if we want to have a comparison that says these structures are present for all cells to carry out their basic functions. Right? Now, does that mean there might be a few that are unique to plants and unique to animals? Yes. Okay, but they are going to have some that are going to be common. All right, so what we're doing is we are looking at different types of cells in this lab. Okay. The thing we're looking for is their structures. Okay, and specifically, we're looking for common structures. Because I would say that a reasonable assumption here would be that any structures that are present in all of the cells would be necessary for them to carry out their basic functions. So we're looking for common structures for that reason. In order to be fair, or controlled, we are only looking at eukaryotic cells. Because only eukaryotic cells have compartmentalization. Okay. All of the cells are going to be viewed under high power. Okay. Your final picture of each one of these specimens must be on high power. And I'm sure you can come up with one more thing that might be in common for all of the things we do in that lab. Okay. For our hypothesis. The if part needs to answer the problem, okay? The whole purpose of doing this lab is to figure out what things are necessary for a cell to carry out its basic functions. So I would say the if part should probably talk about the second point of the cell theory. Because if that's what cells do, and we look at them under a microscope, then I would say you should probably list a few of the structures that are going to be visible. And maybe why. So if you were to say then a nucleus will be visible in all of the cells, you'd probably want to tell me that why the nucleus would be there. I would say list a few. Okay? And again, list ones that are going to be visible. Ribosomes are there, but we're not going to see them. So the order we're going to do that in is plant, animal, and then cheek. And then you can look at blood when you're done that. Okay. It says toothpick on here. I can't remember if I changed that on yours or not. It's not toothpick anymore. I used to have people scrape the inside of their cheek with the toothpick, but I had too many people piercing their cheek with the toothpick. And then they were also seeing blood cells on the slide, and that's kind of a big deal because blood can carry disease and stuff like that. So we use the Q-tip now, so no blood ever gets drawn. Okay? So it's just a nice, easy swab. Now, for the purpose of that whole COVID thing and health and safety and all that kind of stuff, you do your cheek. Nobody else touches your slide or your. So don't share the Q-tip. Okay. I don't care how much you like that person, okay? Don't use the same Q-tip, okay? Don't use the same slide, and when you're done, make sure everything gets wiped down and the slide gets washed with soap and water in the sink and then dried off and returned, okay? Because it is gonna have some stuff from you on it, right? And, if you're sick or something like that, then it could be transmitted, and we don't want that. Okay, so those will be the things we'll be looking at. And then for your analysis, there's only one question. But I'm looking for a paragraph answer. Okay, so it says, based on your knowledge of cell structure and your observations of different cells in this lab, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? Explain. Okay, so this is where you would tell me, every cell I looked at had a nucleus because the nucleus does blah, blah, blah. Okay, and you do that for any other structures that are common. And then if you saw some structures only in one type of cell, you could assume that they were probably important for that cell's mode of nutrition. Would it be a good idea to use the proper terms for those modes of nutrition in your answer? Yes. Okay, so I'm looking for a paragraph here, and not two sentences. That's not a paragraph. Okay, I don't, was it 11 points? I'm not going to know whether you do an 11-point paragraph. I don't care either. Okay, I just want you to have the stuff in there that tells me, Mr. Codere, I understand these were the things that were visible. I know what they do, and that means that they're important for this. Okay. Then for your conclusion, copy and paste your hypothesis. Tell me whether you saw the things you predicted. Why did you see them? Why didn't you see them? Can you accept or reject your hypothesis? And then sources of error. You may not use inexperience a second time. You are now experienced because you used a microscope and you're going to be using it again. Okay, so come up with a couple of things. The other source of error you figured out could be useful still. Okay, we are, after all, using prepared slides. Okay. So you could use that. All right, questions there? Okay, so Tuesday morning, we will just go, I'll quickly remind you of a few of these things and then we'll go across and get going on looking at the slides. And you will not have a quiz on Tuesday because it's the first day back for that week. All right, you still have about 15 minutes or so. I don't have the Chromebooks, but you could be working on your phone. You can get some of this done or some of your microscopy lab or your toxin project or something like that. Okay, well, let's make good use of our time here. No worries. We just went over the other bits about cell transport. And you good? Mm-hmm.